Welcome to video 11 of our IB Biology course. Here we are looking still at topic 2, molecular biology, and we are looking at carbohydrates and lipids. That's unit 2.3. Okay, so before we get going here, we just need to have a bit of a background understanding on our organic and inorganic compounds. Okay, so the four most common elements found in all living organisms are carbon, hydrogen, oxygen, and nitrogen. And on our periodic table here, we know that these are all elements. Got our group 1 there of hydrogen, and over here we've got our carbon, nitrogen, and oxygen, all of which are non-metals. Now, when we talk about organic compounds, we're talking about these complex compounds made of carbon found in living organisms. Now, they always contain carbon, hydrogen, and oxygen, sometimes have other compounds, other elements, compounds attached to them. Now, conversely, we have our inorganic compounds, which are much more simple carbon-containing compounds, such as carbon dioxide, or carbonates and hydrogen carbonates. Okay, so much more simple molecules, still containing carbon, inorganic, to our complex compounds containing carbon, also containing hydrogen and oxygen, being those organic compounds. So the diagrams there below the periodic table, you've got that complex structure on the left and a much more simple structure on the right, so the right being the inorganic, the left being the organic compound.
Molecules make you think of chemistry, right? Well, they're also very important in biology. In this video, we're going to look at carbohydrates, proteins, and lipids. When I say lipids, I'm talking about fats and oils. Life processes depend on biological molecules, whose structure is related to their function. Did you know that living organisms are actually made from quite a small group of molecules? What do you think the most common molecule in you is? It's actually water. About 55 to 60% of an adult human's body weight comes from water. And in some organisms, this can be up to 90%. Your muscles and kidneys are particularly watery, at about 79%. Even your bones are made up of 31% water. Bet you didn't expect that. Okay, enough of water for now. Imagine we removed all the water in your body. What would be left? Well, it would pretty much be just proteins, lipids, carbohydrates, and nucleic acids such as DNA. So what are these all made of? Well, carbohydrates and lipids are really simple. They just contain 3 elements: carbon, hydrogen, and oxygen. Proteins are a little more complex, with 4 or 5 elements: carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Let's start by looking at carbohydrates. There are 3 main types of carbohydrate: monosaccharides, disaccharides, and polysaccharides. Simple sugars like glucose and fructose are monosaccharides. Sucrose, which is made up of 2 simple sugars joined together, is a disaccharide. And some carbohydrates are polymers that are made up of lots of sugars joined together, and so are called polysaccharides. Starch, glycogen, and cellulose are 3 important polysaccharides. Cellulose is found in plant balls, and starch is the storage unit for surplus glucose made in photosynthesis. Glycogen is the storage unit used by animals for surplus glucose, and is found in the liver and muscle tissues. Starch is tested for using an iodine solution. If the solution turns blue-black, then starch is present. Let's end with lipids. Many lipids are triglycerides. These have a single glycerol molecule with 3 fatty acid tails attached. Different lipids contain different fatty acids. Triglycerides are a form of dietary fat found in meats, dairy, and cooking oils. The liver also makes triglycerides. They are used by cells and tissue for energy, or otherwise stored as fat. Some examples of lipids are steroid hormones, cholesterol, vitamin A, and vitamin D.
Okay, so after looking at that, let's look closer at our carbohydrates and our lipids. Okay, so carbohydrates, as the name suggests, carbohydrates only contain carbon, hydrogen, and oxygen. And you can see a molecule there. Carbohydrates are the most abundant molecule in living organisms. They're a source of energy in both plants and animals. So remember our chemical reaction of respiration, we need glucose and oxygen as our reactants, okay? In the process of photosynthesis, where we say that plants make their own food as such, your reactants are water and carbon dioxide, and these react to give us the product of glucose and oxygen, that glucose then being what they use as their energy source. So carbohydrates are our energy sources, what's required in respiration. Now, they function primarily as a short-term energy source. They have an important structural role in plants, so that's cellulose in the cell wall. That is made out of a carbohydrate, and you can see in the image here, we've got our glucose monomers all connected together, okay, to make our long cellulose strands. They occur in many different forms. So we can see here we've got some glucose, that's a monosaccharide. We've got sucrose, disaccharide, polysaccharide. Here we've got amylose starch. Okay, so we've got our monosaccharides, which contain one subunit. So mono, one sugar. We've got disaccharides containing two subunits. So di, two. Saccharide sugar, two sugars. Examples of those are maltose, lactose, sucrose. And then we've got our polysaccharides, poly meaning many, sugar meaning that sugar unit. So some examples of these: cellulose, glycogen, starch. So here we've got our three types: carbohydrate, monosaccharide, disaccharide, polysaccharides. We've got our examples, how they're used in plants, how they're used in animals. Alrighty, so when we've got our monosaccharides and they join together to form a disaccharide, water is released. It's what we call a condensation reaction. So condensation being the release of water. Okay, so if we look here, when the molecules join, they join across the oxygen, releasing two hydrogens and an oxygen as water. So it's a condensation reaction. All right, so one of your understandings here, monosaccharide monomers are linked together by condensation reactions. We've just said that. And then they form disaccharides. And also polysaccharides are also made the same through condensation reactions, because you've got more saccharide units, whether it's monosaccharides, disaccharides joining together. So as soon as those saccharide monomers bond together, we get a condensation reaction. So looking closer at our condensation reaction, okay, so condensation reaction occurs when two smaller molecules join to form a larger molecule held together by a strong covalent bond. So remembering our covalent bond, here it's a glycosidic bond, but a covalent bond is a bond between nonmetals. Each reaction requires an enzyme to catalyze the process, and it results in the production of that water molecule. So here in the diagram, we're looking at glucose and fructose joining together to form sucrose. This type of reaction occurs to form disaccharides from monosaccharides and polysaccharides from smaller units. Okay, so if we think of this building of our polymers, we look at maltose. Maltose is a disaccharide. So we've got glucose, two glucose units here. So two glucose molecules, sorry. Our condensation reaction forming our maltose molecule here and water. Lactose, we've got galactose and glucose joining to form our lactose. Now in our condensation reaction where we are forming the lactose, water is released. However, if you look here, we're going from lactose back to its monomer units, and that's actually what we call hydrolyzing. Okay, so water is required to separate those two units out. Sucrose. So we've got glucose bonding with fructose, and that then gives us sucrose. So just in summary there, we had maltose, sucrose and lactose, all of which are disaccharides, but the way they're different is their monomer sugars are different. So maltose is the two glucose molecules, sucrose is glucose and fructose, and lactose is galactose and glucose. And when they form during a condensation reaction, water is also released. Application 1. The structure and function of cellulose and starch in plants and glycogen in humans. So you need to have an understanding of the structure of cellulose in plants and starch in glycogen and starch in both animals and plants and glycogen in humans. Okay, so polysaccharides of glucose. So that's basically what that application is asking. So first of all, we've got starch. Now, starch is an energy storage polysaccharide found in plants. It is composed of alpha glucose units and exists in one of two forms. We can have amylose or we can have amylopectin. Okay, so you see amylose is more of a straighter chain, whereas amylopectin branches off more. In starch, the arrangement of glucose molecules can be linear, like in amylose, so in that case called amylose, or branched, like in amylopectin. Amylose contains 1 to 4 bonds between the glucose molecules, whereas amylopectin contains an additional 1 to 6 bonds. Okay, so here we are looking at cellulose. Now, cellulose is a structural polysaccharide that is found in the cell wall of plants. Cellulose is made up of beta-glucose subunits with 1 to 4 bonds between the glucose monomers, making it a linear shape. Okay, so here we're looking at glycogen. And glycogen again is a polysaccharide. This time it's used for energy storage formed in the liver in animals. It's composed of alpha glucose subunits linked together, making it branched, and contains 1 to 4 and 1 to 6 bonds between the glucose molecules. Alrighty, now we looked at condensation reactions. Now we're going to have a look at hydrolysis reactions. Now, of course, these are, as I said earlier, the opposite of condensation reactions. They tend to occur when food is digested. Okay, they require an input of oxygen and an enzyme to catalyze the reaction. Hydrolysis breaks down polysaccharides into monosaccharides. Hydrolysis breaks down triglycerides into glycerol and its 3 fatty acids. So hydrolysis is that breakdown. All right, one of the skills of this unit is to have the use of molecular visualization software to compare cellulose, starch and glycogen. Now we will be doing more of this in class, but just to show you in advance. So this is what we mean by molecular visualization software. So you're getting that 3D image of the molecules we're looking at. So here you can see cellulose, glycogen, amylose, and amylopectin. Amylopectin. Okay, understanding 4. Triglycerides are formed by condensation reactions from 3 fatty acids and 1 glycerol. So remember that condensation reaction is the building up of a molecule where water is released.
Lipids are a group of hydrophobic biomolecules that play important roles in living organisms. While the primary function of lipids is long-term energy storage, lipids are also used for protection, insulation, and lubrication. They also act as precursors for some hormones and are a key component of cell membranes. There are 4 basic groups of lipids. These are triglycerides, phospholipids, steroids, and waxes. Although these groups differ in many respects, they all have one characteristic in common. They are all insoluble in water. You may have noticed that lipids and water do not mix. For example, notice the yellow-colored oil in the beaker of water shown here? Even if we stirred this for several minutes, or even several hours, the oil would still separate out from the water. This is because lipids are hydrophobic. From Latin, the prefix hydro means water, and phobic means fear of. So when you hear that lipids are hydrophobic, this basically means that water and lipids do not mix. Let's take a closer look at the category of lipids known as triglycerides. Triglycerides include the fats and oils. Fats, such as lard and butter, are solid at room temperature and are used by animals for insulation, protection, and long-term energy storage. Oils, such as corn oil and olive oil, are liquid at room temperature and are used by plants for long-term energy storage. At the molecular level, triglycerides contain 2 types of subunit molecules, glycerol and fatty acids. Let's take a quick look at fatty acids. A fatty acid has 3 main parts: a chain of carbon and hydrogen atoms called a hydrocarbon chain, a methyl group at one end, and an acid group at the other end. Fatty acids can either be saturated or unsaturated. A fatty acid that has only single carbon-to-carbon bonds is known as a saturated fatty acid. This is because the carbon chain is saturated with all the hydrogen atoms it can hold. Unsaturated fatty acids have 1 to several double bonds. Double bonds result in kinks in the fatty acid chain, which affects the melting point of the fat. Animal fats have saturated fatty acids and are solid at room temperature, while vegetable oils have 1 or more double bonds and are liquid at room temperature. A trans fat is an example of an unsaturated fatty acid where the hydrogen atoms are on opposite sides of the double bond. Trans fats are usually formed during the production of processed foods and are also common in partially hydrogenated oils. In order to increase shelf life and melting point of the fat, excess hydrogen atoms are introduced to an unsaturated oil. This causes the formation of trans fat bonds in the fatty acid chain. Unfortunately, the consumption of trans fats has been associated with cardiovascular disease, and its use has fallen from favor. Now that you understand a little bit about fatty acids, let's zoom back out and look at how the triglyceride subunits fit together. Remember, a fatty acid is only a small part of a triglyceride. To become a triglyceride, 3 separate fatty acids have to bond with a glycerol molecule through the process of dehydration synthesis. Let's move on to the next category of lipids, which is phospholipids. Phospholipids are similar to triglycerides in that they contain glycerol and 2 fatty acids. What's different is that a phosphate group, rather than a 3rd fatty acid, is attached to the 3rd carbon of glycerol. Phospholipids are extremely important, mainly because of their unique properties in regard to water. The phosphate head of the molecule is hydrophilic, or water-loving. This means that it mixes well with water. The fatty acid tails, however, are hydrophobic, or water-hating, and do not mix well with water. Because of these unique properties, phospholipids tend to arrange themselves so that only the hydrophilic heads interact with the watery environment, and the hydrophobic tails crowd inward away from the water. This structure is the major component of plasma membranes of the cell. Steroids are the next category of lipids. Steroids are composed of 4 fused rings of carbon, to which different functional groups are attached. One well-known steroid molecule is cholesterol. Cholesterol serves as a precursor for the synthesis of other steroids such as testosterone, estrogen, vitamin D, and cortisol. Cholesterol is present in plasma membranes where it stabilizes the membrane. The hormones testosterone and estrogen have small differences in their functional groups, but large differences in their effects on an organism. Waxes are the final group of lipids. Waxes are nonpolar and repel water. They're found in the protective coating on leaves and on outer surfaces of animals. Wax is produced in the ears of some animals to protect the eardrum. In addition, bees construct honeycombs from wax. Now that we've covered all 4 categories of lipids, let's do a quick recap. The 4 categories of lipids are triglycerides, phospholipids, steroids, and waxes. All lipids are insoluble in water. While the primary function of lipids is long-term energy storage, lipids are also used for a multitude of other purposes, such as protection and insulation, and as a key component of hormones and cell membranes.
Okay, so looking a bit further there at lipids after watching that video, we can see here we've got our glycerol and our 3 fatty acid molecules, and through that condensation reaction we achieve our triglyceride there, or we obtain a diglyceride. So again, condensation reactions are required for a diglyceride to form. 3 fatty acids join in the glycerol and water is produced. 1 water molecule will be produced for each of the 3 fatty acid tails that attach. Each hydroxide group in the glycerol is able to combine with the carboxyl group of the fatty acid. First application, lipids are more suitable for long-term energy storage in humans than carbohydrates. Why is this? Okay, so lipids include fats, oils, waxes, phospholipids, glycolipids, and steroids. They're more suitable for this long-term storage of energy in humans than carbohydrates. And this is because the amount of energy released in cellular respiration of 1 gram of lipid is double the amount that is released through 1 gram of carbohydrate. However, carbohydrates are more easily digested, so are the major energy source. So lipids are more efficient and produce more energy, but carbohydrates are easier to be digested, and therefore that's the major energy source. Lipids are less dense than water, so fat storage helps with large aquatic animals to float. Structural components of membranes are phospholipids, and non-polar insoluble molecules, so they don't affect the movement of water in and out of a cell by osmosis. They're important in providing heat insulation. Okay, fat storage under the skin helps to prevent heat loss and are vital for animals living in cold conditions, for example, polar bears and sea. Understanding 3. Fatty acids can be saturated, monounsaturated, or polyunsaturated. What do we mean by saturated? So fats and oils of plants and animals are triglycerides. So we spoke about that. We spoke about that molecular structure of our triglyceride. Okay, so fats in animals are solid because their fatty acids are what we call saturated, making them straight. packed very close together. Okay, oils in plants are what we call unsaturated, and this is because they contain double bonds to form bends or kinks, so they cannot pack close together, therefore they form liquids. Okay, so we've got our unsaturated molecule here and a saturated molecule, our saturated molecule here, which is that straight molecule, and unsaturated molecule here, which has those double bends forming those kinks, meaning it can't compact closely together, so unsaturated being those liquids. If there is one double bond, the fatty acid is mono unsaturated because there's only one bond. If it has more than the one double bond, then we say it's polyunsaturated. Okay, so our saturated fatty acids here found in animals, remember, because they're straight, unsaturated, they're saturated, they're straight, they can be packed closely together. That means they're solid. Here we've got it unsaturated, so they've got these kinks in them. However, monounsaturated means it's got one kink, so there's one set of double bonds in there, whereas our polyunsaturated has several double bond sections, meaning there's several kinks in it. So it's not just the one bend as such, there's several. Okay, saturated, mono, unsaturated, or polyunsaturated. Caproic acid, there is no double bond in here. It is saturated. Saturated, monounsaturated, or polyunsaturated. We've got one double bond there, making it monounsaturated. That's our oleic acid. And here we've got linoleic acid, and that is polyunsaturated because it's got several double bonds. Understanding 3, unsaturated fatty acids can be called cis or trans isomers. So we've got our two diagrams there. Unsaturated fatty acids can either be cis or trans isomers depending on the position of the two hydrogen atoms around the carbon-to-carbon bond. So there we've got the carbon-to-carbon bond. Cis, the hydrogen are on the same side. Trans, they're on different sides. So here we've got our oleic acid, and our hydrogen molecules are then on different sides, so that's our trans, and our oleic acid, our hydrogen atoms would be on the same side, so that's why oleic acid is a cis. Application 2, scientific evidence for health risks of trans fats and saturated fatty acids. And application 4, evaluation of evidence and the methods used to obtain the evidence for health claims about lipids. There's been claims about the effects of different types of fat on human health over time. The main concern is coronary heart disease, in which arteries become partially blocked by fat deposits, leading to the formation of a clot. Now, there is a positive correlation that's been found between saturated fat intake and coronary heart disease. However, does this prove that saturated fats cause coronary heart disease? Could it be another factor correlated with saturated fat intake, such as low amounts of dietary fibre, that actually causes the coronary heart disease? A positive correlation has also been found between trans fat intake and coronary heart disease. Other risk factors have been tested to see if they can account for the correlation, but none did. Trans fats may therefore cause coronary heart disease. What other evidence could be used to support this claim? Skill 2, determination of body mass index by calculation or use of a nomogram. So here we're looking at BMI. This is where we look at body mass index. It's a measure of the body fat based on height and weight. It can be used as an indicator of high body fatness. However, it does not provide a diagnosis of body fatness or the health of an individual. Okay, so that's the diagram there. So if you've got a BMI of 18.5 to 24.9, then you're classed as normal and so on. But it's not an accurate measure of the person's health and fitness. The BMI of a person can be calculated by dividing the weight in kilograms by the square of the height in meters. And then here we've got our table showing that. Okay, now an alternative to calculating the BMI is using a nomogram that we've got here. Simply use a ruler to draw a line between the weight and the height of the person, and in between you'll cross where they are in terms of body mass index. Thanks for watching that video. I know this one was a bit longer, and I will see you in the next one.